All right, everyone. Welcome back to virtual BI207 lab. This week we're going to be talking about special senses. So talking about some eye and ear anatomy, and a little bit of physiology as well. This is your last lab. So this week you will have special senses stuff due, and a quiz over special senses. And then next week is your lab final. So we'll talk about the final in a second. But first, I just want to point out on your special senses handout, if you look at that, it says at the top the homework is optional. The homework is not optional for special senses. So typically, if this is the last week of class, we wouldn't have a chance to check your homework the following week because it's the final. But since now we're completely online, all of your participation points are coming from homework, and you can submit the pictures online, obviously. So please do the special senses homework and submit it on the Dropbox by the scheduled due date. So the homework is not optional. You are required to do special senses homework. If you have questions about that, let me know, but I think that's pretty clear. Okay, so before we talk about special senses, I want to just discuss the final really quick and what to expect. So it's obviously going to be online, which is a little bit weird. Um, maybe easier, maybe harder. I'm not really too sure what to expect for you guys. But it's going to be the same as the midterms. So it's going to be 50 points, and you're going to have a 2-hour time limit, just like you would if you were actually in class taking it. The final, it's really important to note that the open and close dates are going to be different than what it's been for all of the labs. So we've had the labs open Monday at 8 a.m. and they close Sunday at 11:55 p.m. The final is different, okay? So the final is going to open Tuesday, April 21st at noon and close Thursday, April 23rd at noon. So you have all afternoon Tuesday, all day Wednesday, and then Thursday morning to complete the final. The reason we're doing this during those times is when the labs are supposed to be scheduled. So instead of having it open for such a long time, we wanted to make sure it was just open during the times you would actually be in lab. This way, if there's any issues, it's easier. Everyone's kind of taking it closer together. It just makes it easier for us to monitor how everyone's doing. And it's during the time you would normally have class anyway. So whenever you want to take it during the time it's open is fine. You do only have 2 hours, which should be more than enough time. But it does close at noon on Thursday. So if you started at 11:30 on Thursday, you'll only have 30 minutes, right? So no matter what time you start, it's closing at noon on Thursday. So if you wait till the very last minute, it'll shut you out right at noon. So make sure you start early enough that you're done with your 2-hour time limit by noon. Hopefully that makes sense. There should be no scheduling issues because your classes are normally happening in this time. If for some reason there's an issue, please let your lab TA know as soon as possible. If you can't take the final for some reason during these times, you better have a good excuse. If your lab TA decides, they might be able to open it for you at a different time, but there really should not be an issue with taking it during these times. So please plan accordingly. Don't wait to the last minute to take it. If you have any issues, if you're kicked out for some reason or you lose Wi-Fi, whatever it may be, you need to email your lab TA immediately if you need assistance with anything for the final. So yeah, I guess that's all I have to say. The final is just going to be covering the material from the midterm on. So the first half of the semester you won't see. So the final will just cover the muscle anatomy, muscle actions, muscle physiology. nervous system 1, so the brain and cranial nerves, nervous system 2, the spinal cord and spinal nerves and the reflex arc, and then special senses. So 50 points, it's going to probably just be 50 1-point questions. So it should be pretty easy. 2 hours should be more than enough time to complete that. I would recommend, if you don't know an answer and you plan on coming back to it, there's the option on Educat to like flag questions. You can go back to the questions that you have flagged. I would recommend the first time that you go through the whole exam though, even if you don't know the answer and are going to flag it and come back to it, at least put something in, right? Make your best guess at that point when you have the question open, and then flag it so that when you're done, you can come back to it. That way, if you run out of time, at least you attempted the question and it's not just completely blank. So if you type in an answer or pick a multiple choice answer and flag it and come back to it, you can obviously change it. So please at least put something in, so that way if you run out of time or if something happens, you have some issues, whatever, at least you have some answer saved. Also on Educat, there is the extra credit quiz for the final. So this is exactly the same as the midterm extra credit. It's 5 points. It's 50 questions. You can take it as many times as you want. And if you get all the questions correct, you get 5 points of extra credit. I forget exactly what day this is closing. I think it's closing on Thursday, the 23rd at noon as well. But double check on the quiz, it'll tell you what day it closes. So and just your last opportunity for extra credit for lab. And then one more announcement just before we start the lecture today. Make sure you go through and check through your Educat and the grades on Educat, and make sure everything looks okay. If there's any issues, it helps if you notice them now and email your TA about them now, so we can go in and figure out if there was a mistake grading or something like that. As we get closer to the end of the semester, things are becoming more and more hectic. So just go through and double check to make sure that your grade book looks okay. And if there are issues, email us as soon as you can so we can resolve those. Okay, I think that's all the announcements I have. If you have questions, feel free to email your TA, or you can email me, Claire. I'm the lab coordinator, so I can answer any questions that you have. Also, Giuseppe, Dr. Cortese can answer questions as well if you have them. So, all right, let's talk about some special senses. So besides this lecture that I'm recording today, there's also a video that's going to be posted of a sheep eyeball dissection, I believe, or cow eyeball. I can't remember which animal, but an eyeball dissection that Ryan did. Normally we do that activity in class, and it's kind of cool just to see the different parts of the eye and an actual eye. So take a look at that. You're not going to be quizzed or have an exam that has like pictures of an actual eyeball dissection, but I think it's kind of cool and helps you kind of visualize what the eye looks like better than just diagrams would. So take a look at that. Okay, we're going to start with some ear anatomy. So this is what an ear looks like, obviously. So the ear can be divided into 3 parts, depending on where you're at. Of the outer ear, middle ear, and then the inner ear. Okay, and each part of the ear obviously has multiple structures that make it up. So the outer ear is made up of the pinna, which is this outside part of the ear that you can see. You should know what type of cartilage this is made of. If I was in class, I would ask you this right now, but you guys can't talk to me. But this is elastic cartilage, right? So really flexible cartilage. And this just functions to collect sound waves from your external environment and bring them into your ear. Okay, it brings them into your ear through the external acoustic meatus, also known as the auditory canal. So if you remember back to the skeleton, we learned about the external acoustic or auditory meatus, and then we had the internal acoustic or auditory meatus, right? So you could see the bone here. So that hole in the skull is where your ear canal, auditory canal, is going to go through. So sound waves get grabbed by the pinna, and diverted down through the auditory canal. The most inner part of our outer ear is the tympanic membrane, so this little guy here. Also, common term for that is the eardrum. In an anatomy class, though, you're expected to know tympanic membrane. So this tympanic membrane is the eardrum. So that's the most internal part of your outer ear. If we go in further, then we enter our middle ear. So all of this is your middle ear. So your middle ear is composed mainly just of 3 ossicles. And an ossicle is just a really small bone. So you have 1, 2, 3 little tiny bones in your middle ear. Collectively, they're known as the ossicles. So you have the hammer or malleus that is connected to your tympanic membrane. You have your anvil or incus that's in the middle. And then you have your stirrup or stapes. And your stirrup, as you can see here, is what attaches to your inner ear. Right, so this whole snail structure is your inner ear. So your stirrup, you can see this like oval shape of the bone. That actually is going to connect to the oval window. So on your inner ear, there's this little window that the stapes or stirrup connects to. Either one of these names for all the ossicles is fine, whatever you remember best. Okay, so the inner ear, we have our oval window, our cochlea, which is this snail-shaped structure here. We have our vestibule, which is this kind of V shape up here. At the top of the vestibule, we have 3 semicircular canals, which are really important for balancing. And then at the bottom below our oval window, we have our round window. And then leaving our inner ear, we have our vestibular cochlear nerve. It gets its name because it comes from the cochlea, right, the snail shell part, and the vestibule. So the vestibular cochlear nerve is cranial nerve number 8. You should know that by now. So it carries auditory information as well as like balance equilibrium information. So the inner ear is actually filled with fluid, and that's how we can hear things, as well as how we know like where our head is positioned in space. So the cochlea is where hearing is taking place, and the rest of the inner ear is involved in balance. Okay. And then the last structure here that's kind of interesting is our pharyngotympanic tube, also known as the auditory tube. So this connects our middle ear to our nasal cavity. And the pharyngotympanic tube is important in making sure that the pressure inside of our ear is equal to the atmospheric pressure. So if you ever like on an airplane or like driving up or down a hill or something, your ears pop. That is basically your middle ear coming into equilibrium pressure with the external environment. And it does this through the pharyngotympanic tube. So if you have a clogged nose, something like that, or it can clog this tube up, and then you can have your ears feel like really pressury because they aren't in equilibrium with your atmospheric pressure. Okay, so that is the main anatomy of the ear. If we zoom in a little bit closer on that inner ear, so again, we have the cochlea, our vestibule, and our semicircular canals. There's a few additional structures that we have to know. So at the bottom of each semicircular canal, there's a kind of a little bulge that is called an ampulla. So all these 3 little bulges, this back one's hard to see, are ampulla. And then our vestibule can be divided into 2 parts. So the top part of our vestibule is called the utricle up here, and this bottom part is called the saccule. So together, this big V part, again, is the vestibule. Top part utricle, bottom part saccule. Okay. You can see, again, our vestibular cochlear nerve, and cochlear nerve there. Okay. So a little bit of physiology, how hearing and sound works. So basically, again, sound waves are going to come in, get captured by our pinna, and put through our auditory canal. So sound waves are like physical, waves, right? So the waves hit the tympanic membrane, and the movement of that tympanic membrane causes our 3 ossicles to move as well. So every time a sound wave hits this tympanic membrane, it causes these ossicles to move. The stapes, again, is the ossicle that's connected to the oval window on our inner ear. So when the stapes moves, it bounces on that oval window, and our inner ear is actually all filled with fluid. So when that stapes bounces on that window, it causes physical waves within that fluid in our inner ear. So it goes from like sound waves in the air to a mechanical force moving these ossicles to like liquid waves within our inner ear. And then our cochlea, you might talk about in lecture. We're not going to get too in depth for this in lab. But our cochlea has what's called little hair cells that are the receptors which allow us to actually hear things, turn the waves into action potentials that our brain can interpret. So the hair cells in the cochlea, basically the waves come in and knock those hair cells over, and that knocking over of the hair cells creates an action potential, and then that information gets sent to the brain through the cochlear nerve, vestibular cochlear nerve. Okay. And so again, in lab, we're not really going to ask questions on that. It's more focused on anatomy, but it's kind of interesting to see how it all works. We are going to talk about different types of hearing loss, though. So hearing loss, obviously, there's some sort of issue that's causing you not to be able to hear the sound waves as well as you should. So there's two types of hearing loss that you need to know. So the first is conductive hearing loss, and conductive hearing loss occurs when sound waves can't make it to this inner ear. So if you have some sort of blockage in your auditory canal, if your tympanic membrane is damaged, if your ossicles are damaged, and those sound waves can't even get into the ear, that causes conductive hearing loss. So there's cases where the ossicles will calcify, so they can't move as well, or the tympanic membrane gets damaged. Some sort of issue in this outer or middle ear where sound waves can't even make it to the inner ear is conductive hearing loss. Okay, so outer or middle ear deafness is also known as conductive hearing loss. All right, the second type of hearing loss is sensory neural hearing loss. And the name kind of explains where the issue is. The issue is in the actual sensory organ or the neural area. So again, remember I said the hair cells in the cochlea are the receptors that are actually going to transmit the sound information to an action potential for the brain to interpret. So if there's damage to those hair cells within the cochlea or damage to the nerve that's taking auditory information to the brain, then we have sensory neural hearing loss. Right, the sound waves are coming in just fine, creating waves in the cochlea just fine, but for some reason, either the hair cells are damaged in the cochlea or our vestibular cochlear nerve is damaged, so the sensory information can't make it to the brain. Okay, so that would be inner ear deafness or sensory neural hearing loss. All right, and normally in lab, we do different activities to show the difference and test for conductive versus sensory neural hearing loss. So there is a YouTube video. I linked it on Educat as well. I'm not going to play it in this video just for time's sake, but it shows the two different types of hearing tests, which are on the next page. We have Renee's test and Weber's test that test for different types of hearing loss. So I'm just going to tell you to watch that video on YouTube. It explains Renee versus Weber's tests, how they're performed, and what they're testing for. So it's really important that you know the different types of tests, how they're performed, and then what type of hearing loss that they're testing for. Again, just watch that YouTube video. The link's on Educat. It explains it better than I can, so watch that. Okay, so that's all about the ear. The next part of the special senses is eye anatomy. So to start, there are a few eye muscles that you have to know. Actually, there is 6 eye muscles. So these are the muscles that are involved in controlling the movements of the eye. So if you're looking a certain way, watching, I don't know, some guy run across the street, our ability to follow the person running, whatever it may be, is due to these eye muscles controlling our eye, right? And if we have conscious control over where we're looking, that tells you what type of muscle this is made out of. So if you remember back, there's 3 types of muscle. We have cardiac muscle, which obviously is not what this is made out of, smooth muscle, and skeletal muscle. So if you remember, smooth muscle is involuntary, skeletal muscle is voluntary. So these muscles are all skeletal muscle because we have conscious control over them. So there's 6 of them you have to know. I'm going to go to the next slide to show you. So you have 4 rectus muscles, which are these 4 straight ones on either side of the eye. So you have your superior rectus and your inferior rectus, your medial rectus, which would be close to your nose, and your lateral rectus, which would be close to your ear. Okay, so the rectus muscles in this picture, these are the straight ones. So there's 4 rectus muscles, just anatomical terms where they're at. And then we have 2 oblique muscles, which are these guys that are going to kind of squidge, move. I don't know. They're like kinked. They're squiggly. That's the word. Squidged. What the hell squidged? They're squiggly. So we have the superior oblique on the top, this squiggly one up here, and then the bottom we have the inferior oblique. Okay, so 4 skeletal muscles that are rectus muscles control just side to side and up and down movement. 2 oblique muscles, again, also skeletal muscles that control the rotation of the eye. Getting into actual eyeball anatomy, our eye has a few different layers. So most superficially, the white part of our eye is called the sclera. So that is the white part on the outside of our eye. Over the front of our eye, we have the cornea. So that's the clear part. Technically, that is continuous with the sclera. It's one piece of tissue, but it just is clear, obviously, so light can get through and so we can see. But the most superficial part of the eye is the sclera or cornea over the front. We have next deep to that is the choroid layer, which is this red layer here. You can see choroid is that middle layer. And then the deepest layer of the eye is the retina. So the retina is what covers the whole inside of the eye. At the back of the retina, we have a spot that is called the fovea, and that is the area that has the best visual acuity. So the retina is composed of the photoreceptors that are going to take visual light information and transmit it into action potentials so that our brain can interpret that information. So the retina has all of those photoreceptors that are taking information from light and converting it to action potentials. The fovea has the highest density of those photoreceptors, right? So when light hits there, it's the clearest image because it has the highest density of those photoreceptors. Leaving the back of the eye, we have our optic nerve. So all of those photoreceptors are just specialized types of sensory receptors. So they all have long axons that are leaving the eye, and those leave in the bundle that is called the optic nerve. And where that optic nerve leaves, we also have some vasculature, different veins and arteries that leave the eye as well. And due to that, that small spot where all that stuff is leaving the back of the eye, we actually have a blind spot. The blind spot is where the optic nerve leaves the eye. And it's a blind spot because there are none of those photoreceptors in that area. Okay. So this image is just showing the retina and then our photoreceptors, which are rods and cones. So hopefully you've all heard of rods and cones before. Cones are important for color vision. Rods are important for monochromatic or like black and white vision, help us see better at night. The physiology of the eye is a little bit complicated and we're not going to get in this in depth in lab. As long as you know the retina is made up of the photoreceptors, which are rods and cones. Okay? All right. More on the eye. So we can see This is the white outside part of the eye, the sclera, cornea, the clear part over the front of the eye. So we have below the cornea, the iris, which is the colored part of your eye. So if people have brown eyes, have this brown iris. You can have green eyes, blue eyes, obviously, right? So the iris is the colored part. Deep to the iris, you have what's called the lens. And you'll see this in the eyeball dissection that Ryan does. It's really cool. But the lens, the function of the lens is to focus light that's coming into the eye. So the lens actually is connected to all these little stringy boys, which are called zonules. And the zonules are connected to the ciliary bodies, which are just little muscles on the outside. So when these muscles contract and relax, it pulls on the lens to change the shape of it to allow light to focus at different points in your eye. Okay, so this image kind of shows that again. Again, the cornea in the front, light's coming in. So you have your iris, the colored part. The hole in the middle of the iris is called your pupil, and directly behind that you have your lens. Okay, so again, the lens changes shape so that the light hits at the perfect point, so it hits the back of your eye and the retina. So again, these little zonules and ciliary bodies change the shape of this lens to focus light. That's how you can see stuff that's up close or focus on stuff that's further away. Okay. Okay. And then the last two things, we have two cavities in our eye. So you can see this front cavity and then this back cavity here. So these are actually empty. Well, not empty, but they're, filled with fluid. So our anterior cavity is in front of our lens, and that's filled with what's called aqueous humor. And that's basically the same as like what a tear would be, right? So really liquidy, watery tear material, I suppose. So aqueous humor is in the anterior cavity. Behind the eye, we have our posterior cavity, which is this big cavity here on the inside of our retina. And this is also fluid filled, but it's filled with something called vitreous humor. And you can see it in the eyeball dissection, so again, you should watch that. But the vitreous humor is kind of like jello almost. It's really like thick consistency, jelly-like. It's a lot more viscous than the aqueous humor. So in the dissection, you should be able to see that, and it's kind of cool. Okay. I think that's it for the eye anatomy. In your handout and in your lab manual, there's an activity that can demonstrate the blind spot. So I would recommend that you do that as well and kind of understand how the blind spot works and why you have a blind spot. Again, it's because where the optic nerve is leaving, you have no photoreceptors in that area. So do that activity, and that's it for special senses. So if you have any questions, let me know. I can try to clarify anything if you need. But yeah, that's that. Okay, so that's your last lab. Make sure you start studying for the final sooner rather than later. Even though you have your notes, it's really important that you are prepared and not just completely relying on your notes. Otherwise, you're gonna run out of time and not have enough time to finish. So get studying. If you have questions, let me or your lab TA know. And yeah, good job this semester, guys. Okay, bye.